Hey guys, my name is Max and welcome to Civil Biology. In this video we're going to be talking about active transport. Now in the last video we talked about passive transport, which is essentially when the cell takes in some sort of substance or for that matter removes it through just simple diffusion, where it just allows the substance to diffuse as it normally would. But sometimes the cell wants to oppose diffusion. It wants to create some sort of high concentration area of that solute. And to do that, it'll need to use energy in the process we called active transport. So that's what we're going to be talking about in this video. Now, like I said, active transport uses energy, and that energy is very important as far as the process goes. It actually comes in the form of ATP, which is, we call it the energy currents of the cell. It's adenosine triphosphate. And the way it works is it'll do some sort of it'll break in order to release energy. That energy will be used to change the shape of the protein, which will then be what actually transports the solute across the membrane. So that's how that works. We'll talk about ATP more in other parts of this unit, so don't worry about it. But that's kind of how it works. And the important thing about active transport is that it actually opposes diffusion. Oh, let me make that a little bit thicker. It actually opposes diffusion. So where diffusion will want to make the solute go from an area of high concentration to low concentration, active transport allows the cell to move solutes from an area of low concentration to an area of high concentration. Now, we call this going against the concentration gradient. A concentration gradient is basically the difference in concentration over some sort of distance. And you can kind of see here we've got 1, 2, 3, 4, 5 pieces of solute. We'll call that maybe a 0.5 molar solution. That's kind of a concentration. And then over here you just have 1, so maybe it's a 0.1 molar solution. And because it's going against the concentration gradient, it's going from that low to a high concentration, this is going to use energy, and it's going to require active transport. Now, if you remember from our passive transport video, we talked about carrier proteins and channel proteins. You notice that you can only have a carrier protein, as we see in this diagram, because what it does is it allows a greater control over what happens to those solutes. The reason for this is because whereas a channel protein will just kind of sit there and allow the substances to go through it, this carrier protein can actually grab that solute and push it through the other side using the energy from ATP like we mentioned earlier. You can kind of see that it's got a little shape specific to the solute, and it'll just change shape to, it'll shuttle the solute across the membrane. And so that's why it would use a carrier protein instead of a channel protein. Now, next we're going to talk about a very specific type of active transport, and this is one that's very important for animal cells. In fact, it's found in every animal cell that we've ever come across. And this is what's called the sodium-potassium pump. What this sodium-potassium pump does is it'll actually pump three sodium molecules out of the cell for every two potassium, not molecules, ions, 3 sodium ions out of the cell for every 2 potassium ions that it transports into the cell. In this diagram, you have the red ions, or the red solute is sodium, and this green one is potassium. And you can kind of see in this diagram, you got the 3 being pumped out and the 2 being pumped in. What you might notice is that there's actually a net charge of -1 inside the cell for each cycle. The reason for this is because if the sodium has a +1 charge and you're having 3 of it pumped out of the cell for each in a cycle, and then 2 potassium pumped into the cell, then this is going to equal 1. But this is outside the cell. We'll call it -1 inside of the cell. So you can see that it is generating some sort of charge. And what this will do is it'll create a membrane potential, which we're going to be talking about in our next slide. Now, membrane potential is essentially a voltage across the membrane. A voltage, if you know, is a separation of charges. So a membrane potential is when there's some sort of separation of charges across a membrane. Maybe one side of the membrane is a more positive side, and then the other side of the membrane is more negative. Generally speaking, because of that sodium-potassium pump, the inside of a cell will be more negative than the outside of a cell. In addition, we have what we call ion pumps. Now, an ion pump is, as the name suggests, any protein that actively pumps or actively transports ions, which are charged particles. So the sodium-potassium pump is a very good example of an ion pump, but it could be just as simple as this diagram. We also call these ion pumps electrogenic pumps because electro meaning like electricity, electro similar to electricity. I'll just draw the electron symbol, and then genic, gen means to form. So it's forming electricity, forming a voltage like we mentioned earlier. And so the electrogenic pump will generate some sort of membrane potential or separation of charges across the membrane. But you also know that since opposite charges attract and like charges repel, so like this positive ion will repel from this positive charge, you know that there's some extra force in there besides just what we have with diffusion. Let me kind of explain that. Since this sodium maybe is positively charged, it'll want to go to an area of negative charge, kind of to neutralize that negative charge. And so there will be some sort of other force that will want to affect the motion of that ion, of that particle. And because of this, we have a new term which we call the electrochemical gradient. So if you remember earlier, the concentration or chemical gradient is the difference in concentration across some distance. You could have an electrical gradient, which is essentially a voltage, a separation of charge, a difference of charges over a distance. So an electrochemical gradient is it factors both concentration and charge. It's the combination of an electrical gradient and a concentration or chemical gradient. And because of this, once you have these two forces factored together, the electrochemical gradient will actually be what determines diffusion. If you remember earlier, we used that the concentration gradient is what will determine diffusion. For charged particles, we actually have to use the electrochemical gradient for a more accurate representation of what's going on. So the electrochemical gradient is actually a better definition of how diffusion will actually work. Now, one last thing, any pump that opposes the electrochemical gradient, earlier we said that any pump that opposes the concentration gradient, any pump that opposes this gradient will use ATP. So perhaps for certain solutes, there might be a chemical or an electric potential electric gradient that is stronger than the chemical gradient. And for these, it would actually go against the concentration gradient, but it's not using any energy. So that would be something to keep in mind, that both of those parts are important. Now, the last thing we have is what we call co-transport. Now, co-transport is when there are two different substances or substrates being transported at the same time. And you'll have one substrate that will actually fall down the concentration gradient. And so this one will not use any energy. But the second one is actually taken against its gradient. So this is a type of active transport that will allow one to go against its concentration gradient without actually using energy for this specific process. Kind of to make it easier for you to see, you've got the red concentration gradient. You know, why don't I do it in red? The red concentration gradient is much bigger, so we got a big arrow, and it's very strong. That red solute will definitely want to go down its concentration gradient. That'll be a very strong force. Whereas maybe for the yellow one, it's not quite so strong. It opposes the red one, but it's just kind of a weak little gradient. The net result will be that it'll all want to go down the concentration gradient in the direction of the red one. One very important example of this is one that we find in plants. And in plants, they'll actually pump hydrogen ions, which maybe we can call these red ones, we can call them hydrogen ions. And it'll pump these hydrogen ions outside of the cell. So there'll be some other protein over here that's pumping the red ones out of the cell. And when it does this, it'll create a gradient of these hydrogen ions, and this will actually be what fuels some other types of transport. One important one that they do is to pump sucrose into the cell. So we're going to call this yellow substance sucrose. And this is what happens in plants when they want to take up sucrose in higher concentrations. It'll use this co-transport. And so that's really it for code transport, and active transport as a whole is just as simple as that.